Hi, everybody. Welcome back to this week's Single Solid Exploration Club, where we demonstrate some of the features on our apps in the form of a case study. So for this week, we're going to be featuring Spatial X because over the past couple of weeks, Spatial X has undergone some changes to the UI. So I definitely want to introduce those changes to tell you guys where you can find all of the tools. And for this week, we'll also be looking at some Merscope Ultra data comparing Alzheimer's brain and normal brain. So this is the agenda for today. I'll give the introduction to the new user interface, and then I'll show you how you can use some of the analytical tools on Spatial X to unlock some insights from the brain tissue. All right, so starting off, let's jump right into Spatial X here. So here, the first thing that you'll notice is that with the new UI revamp, we've definitely optimized the image view because we've recognized the need for the fact that spatial data analysis, the most important thing in that is to visualize the image and to perform some simple gene expression. So we've maximized the width and the height of the image view so that you can get the most optimum experience and more comfortable experience using Spatial X. You notice on the left-hand side, we have our toolbar, which we've squished into this little panel view. But if you hover over the toolbar, you'll immediately expand it and you can see some of the secondary analysis here. Down here is where you can view the regions and the preferences for enabling and disabling each of the layers for this spatial image. So you have your image layer and you have your cell layer where you can enable the cell centers as well as the segmentation if you have it. And you can also enable the transcript layer as well. Okay, so zooming in right now, what we're loading is the segmentation. Oops. So right here, you see the cell boundaries for each of the segmentations. Okay, over here on the bottom right is the tools which you can use to play around with the image. You have the panning, the lasso, the measure. And down here, we also have the help and support where you can access the documentation, some tutorials, our blog, and the interface tour, which will basically give you a tour of all of the things that I'll be going over today. Up here in the upper center, this is your options for your layout. So you can switch to a two-panel view where you can look at two different samples together, like so. Oops, sorry, let me refresh that. Let's go back to another tab. Okay, or you can look at the four-panel view here, where you can look at four different samples. Okay, in the display view, you can also switch to the image view as well as to a 2D view. So this is where you get a single cell view of your 2D or your UMAP here. Next, we have our genes query panel and our metadata panel as well. We've put these together in a more compact view. However, if you think it's too compact for you, you can always expand the views and the columns like so. For the gene query panel, of course, this is where you can use it to query for specific genes, such as this one. Let me enable the cell center. Okay, and click apply. Okay, so this is where you can see the individual cell centers and which cells are expressing that particular gene here. You can also do the same for the transcript layer. So for example, if we enable that, you might be able to get a look at some of these smaller transcripts. Let's take a look at another gene perhaps. And click apply. Okay, and the transcript layer does take a while to load, but you can see something like this, where you can see smaller dots, and the expression of that smaller dot represents the transcript. And since it's within the cell boundary here, that's why the cell center is now highlighted that specific color. Okay, and so that's the gene query panel. And finally, you have the metadata panel. So this is where you can flip through your different type of metadata. For example, this. And you can either view it on your spatial image, or you can view it in the 2D embedding, of course. Okay, and so that covers most of the features for how the new UI is now organized. Again, we wanted to make this huge change because we recognize the need for having more space for viewing your spatial image here. Okay, so let's go back to our slides here. So jumping into our data set for today. So this is our data set for today. These tissue comes from human brain blocks, where one was diagnosed with Alzheimer's and one is a normal healthy tissue. Sequencing with the Merscope Ultra, this is able to give us single cell resolution and a 960 gene panel. So the authors have chosen genes that are specifically used for cell type annotation, as well as marked for neurodegenerative pathways. Okay. And so we recognize that one of the biggest challenges to analyzing spatial data is definitely cell type annotation, not only because this process could be repetitive and it might require some proprietary knowledge about certain marker genes, but also the fact that a lot of spatial data analytical tools use single cell RNA sequencing data as a reference point. And by doing so, this is missing a lot of the spatial context that comes with spatial data. So here I wanted to demonstrate all of the tools which you can use together to help supplement your cell type annotation process. So the first tool here you can use is the simple gene query tool. For example, the authors have chosen SLC17A7 as a marker gene for neural cells. And when we query for that specific gene, you can see in our normal and our Alzheimer's brain that it's pretty localized to a particular layer in the brain, this outer layer here. When we add in a second marker gene, this MOG gene, this one tags for oligodendrocytes in the brain. So you can see how the expression of these two marker genes is pretty distinct from each other, where SLC17A7 is marking for the outer layer of the brain, whereas the MOG is marking for the inner layer of the brain. Okay. Another way you can visualize this distinct separation in these marker genes is through a dot plot or a bubble heat map like this. So the authors have also provided cell type annotations already. So when we look at the expression of MOG, you can see how it's pretty highly expressed or specifically expressed in oligodendrocytes. And for SLC17A7, it's expressed across all of these different cells, but mostly or highest expressed in our neurons. Okay, and so these are the ways in which you can use the gene query if you have some specific marker genes in mind where you want to visualize on your image. But of course, if this is your own data, you will probably not start out with the cell type information. And so now we want to know how we can predict which cell types are included in our samples. And this is where we can use the cell type annotation or the cell type prediction tool, excuse me, on Spatial X. So the cell type prediction tool, basically what it does is it takes from our Talk to Data database, which is our database full of single cell RNA sequencing studies, and it uses the marker genes from those particular studies to compare to the marker genes within our data set and see how well they overlap with each other. So not only is this process pretty automated, but you can also change the final annotation as well, depending on the metrics given within the cell type prediction tool. So the first step to cell type prediction is the clustering. So for clustering, it will cluster together your cells based on how similar their expression profiles are. So you can see that here. And in particular, each cluster would typically stand for one particular cell type. So for each cluster, you can see here how we have a list of the predicted major cell types and a list of the predicted subcell types. And we have other metrics for you to look at the marker genes and to look at the statistics as well, so that you can also change the final annotation. Okay, and so once you're happy with that final annotation, you can click Save to Metadata so that you can visualize it back on your tissue here. In this sample, you can see how the cell type prediction tool annotated pretty well where the neurons and the oligodendrocytes sit. So again, the neurons are found on the outer layer and the oligodendrocytes are found on the inner layer. Okay, another thing that is pretty handy about the cell type prediction tool is within the tool, you can also look at the summary of the studies that were used in the prediction. So for each study or for each data set, you can see how the authors have annotated their particular cluster, as well as the marker genes that mark for that cluster. Okay. And so given that we use the cell type prediction tool to annotate the spatial data, again, remember that we were only using single cell RNA sequencing studies in the prediction. But one thing that was missing was the spatial context. And so this is where we're able to use the region segmentation tool in Spatial X to bring back in that spatial context to help us with cell type annotation. Okay, so essentially how the region segmentation tool works is that it will predict or it will outline particular regions on your spatial image, not only based off of the gene expression information, but also based off of the spatial coordinates as well, where those genes are located. And so given that in the region segmentation results, it was able to predict these 5 regions. For example, if we look at segment 0, it predicts a list of genes, and you can call this list your list of spatially variable genes. This means that these genes, when they are expressed within this particular region, they're expressed at a very similar level to each other. So this segment 0 corresponds to this purple segment here, which is found in the outer part of the brain. And so we can say that this segment 0 could be corresponding to our neurons. And we can take a look at the particular gene list as well to see that we will see some neural cell markers as well. Okay, and finally, we can compare our region segmentation results with our cell type prediction results and see that our segment 0 also overlaps with our neuron. Okay, so in this way, we can use both the cell type prediction tool as well as the region segmentation tool to help us with the cell type annotation. Okay, so next, since we have 2 different conditions that in our data set, Alzheimer's disease and normal, of course, one research question that might come up is what are the differentially expressed genes between our normal and our Alzheimer's, and what genes can we see upregulated or downregulated in our Alzheimer's? This is where we can use the differential expression analysis tool in Spatial X. So here, the results of this will look like this. On the left-hand side, you have your list of differentially expressed genes, where each gene you also have the information for the log2 fold change, as well as the p-value. Down here in the bottom right-hand corner is your volcano plot, which essentially summarizes your list of differentially expressed gene. A volcano plot is also interactive as well. So every dot that you click on will highlight that gene so that you're able to export it. And then up above is our violin plot, where if you select particular genes from the left-hand side, you can see the expression distribution of that gene between your two different conditions. Okay. And so finally, another question that we can ask here is now that we have the cell type annotation results, we want to see how the different cell types are co-localized within our Alzheimer's disease sample. And this is where I want to introduce to you guys one of our newest features which we released, which is called neighborhood analysis. So switching to Spatial X again. So in this neighborhood analysis tool, essentially, again, let's switch to the image view. Again, this will help us answer the question of what kind of cells are kind of co-localized or found with our particular cells that we're interested in. So we know that our neurocells are localized to the outer part of the brain and the oligodendrocytes to the inner part of the brain. So we can ask a further question from here. Well, what other cell types tend to be found in the inner layer versus the outer layer? So in this neighborhood analysis tool, all you would need to provide it with is the cell type metadata. So in the metadata, I will click the cell type prediction or the cell type annotation labels and click run. Click run. Okay, and the results page for this looks like this. So you have two different outputs for neighborhood analysis. The first output is the cell type co-occurrence, where you can see which cell types co-occur with each other. And the second is the neighborhood, so that you can see how many neighborhoods were yielded from this analysis. From this heat map, you can see, for example, our oligodendrocytes don't really associate much with our neurons. And of course, we saw that visually, where the oligodendrocytes and our neurons tend to be found in different layers of the brain. However, if we look at a section like here, where we see our brain pericytes more co-localized or co-occurring with our endothelial cells, right? And this kind of makes more sense because pericytes and endothelial cells play pretty important roles in the blood-brain barrier. So basically, endothelial cells will line the blood vessels of the brain, whereas pericytes will surround those endothelial cells. So we expect these two cells to co-occur or co-localize with each other. Okay, moving over to the neighborhood clustering. So here we get a different type of heat map view. So this is where you can see the amount of predicted spatial domains or an amount of predicted neighborhoods. And within each domain, you can see what type of cell types occur within that domain. So I can move this threshold here to change the clustering resolution of our heat map. Let's say to change it to a resolution of 7 clusters or 7 domains. One thing I can notice from here is, again, the oligodendrocytes and the neurons are found in different domains. Whereas we can see here in our brain pericytes and endothelial cells, this is also found in one particular domain. Okay. Other things we can notice here are that some of these cell types are also pretty specific to certain domains, so they don't particularly occur with other cells, such as this vascular leptomeningeal cell and the astrocytes, for example. Down here, we also have a box plot, which kind of summarizes the proportions of each cell type that you can find in the domain here. So again, our spatial domain 0 has these particular cell types. Okay, and so that was a summary of how the neighborhood analysis feature works here on Spatial X. So let's go back to our slides here. Again, this neighborhood analysis will allow us to answer the question of how different cell types can co-occur with each other. So we saw that with our brain pericytes and our endothelial cells. And we also see how certain spatial domains can have certain cell types co-occur with each other. But I do want to take a moment here to explain that since brain tissue, we see that the layers are pretty well separated, and that the cell types are also well separated. Maybe this might not be the best example of how to use this neighborhood analysis tool to its full potential. So let me go back and find a different tissue for you guys to look at. So maybe if we look at a tissue like lung, for example, where the cell types are not so well separated. All right, let's enable the cell center layer here. And for example, in our lung tissue, if we enable all of the different cell types, and zoom in, you can see that the cell types are pretty scattered, and it's hard to see what kind of cell types would co-occur with each other, except for in important regions like this one, where we see possibly possible scarring tissue occurring here. So we want to see what types of cell types would co-occur in this particular region. And so that's one way the neighborhood analysis tool can help you in this situation. So let's take a look at the results here. Okay, so from here, we can also see that our T cells and our B cells co-occur with each other. So we see a lot of immune cell activity happening within this tissue. Other places where we see some co-occurrence, possibly some fibroblast communicating with the mast cells as well. And if we take a look at our neighborhood clustering, maybe this is where you can see a uh, more of an overview of which spatial neighborhoods these cell types are occurring in. Okay. So again, here we see possibly these smooth muscle cells are pretty specific to a particular domain, but other cell types such as pericytes, endothelial cells, again, macrophages and fibroblasts will occur all in one domain, in one neighborhood. All right. So that concludes today's talk on Spatial X. I believe my colleague here will send out a poll for you guys to answer. For those of you who are new to Bioturing or new to Spatial X, you can also head to our website, bioturing.com/spatialx, to request a demo as well. So basically in this poll, we definitely want to gather feedback from you guys, learn about what types of spatial data you guys have and what types of analyses you guys are looking to perform, and also what kind of talks you guys want to hear in the next club as well. We're also opening up the floor to questions, so feel free to put questions in the Q&A. I'll do my best to answer them. Okay, give it another minute or so for you guys to finish answering the poll and any questions that you guys have as well. Okay, so we have one question from Keisha who asked, does also show cell co-occurrence enrichment within the spatial domains? Show cell co-occurrence enrichment within the spatial domains. So are you in particular talking about how different cell types are enriched in spatial domains, such as this one? So for example, we can see within spatial domain 7, this is pretty enriched in our smooth muscle cells, whereas in spatial domain 4, it's mostly our T cells. Something like this. Proximity of cells within the spatial domain. So that one, I believe, uses a different type of tool called distance query. And I think this is something that I can follow up with you with an email on. So you can also get in touch with one of our other Spatial X product specialists as well. Okay. Tian asks, can you show damaged cells surrounded by immune cells? So yeah, that is something that you can also use, or that you can also ask and answer with using the neighborhood analysis tool. So if you are interested in a particular region, like for example here, and you see a lot of immune cell activity, you might also be able to see some other cells kind of occurring within that hotspot. All right, so if there's no other questions from anyone, any other questions, also feel free to email us at support@bioterrain.com and we'd be happy to answer. So thank you, everyone. Thanks for joining this week.